I'm going to talk, hopefully maybe as you eat your sandwiches, around the topic of randomised trials in a pandemic situation, which in my opinion are an essential rather than a luxury. As a bit of background, I thought it would be just important to start about why do we need randomised trials at all? What are they for? And really, randomised trials are the best way and really the only reliable way to find out about whether medical treatments for any condition, whether those treatments be pills, operations, therapies of any description, whether those are safe and effective. And to do that, we need to compare a group of people who receive that treatment with an appropriate group of controls, so people who don't receive the treatment. But when we're making these sorts of comparisons or doing these experiments, there are two errors that we can make. The first is called systematic error, sometimes also known as bias, and the second is called random error, otherwise known as the play of chance. I'll just explain those in a bit more detail now. So systematic error or bias largely comes when you're trying to work out whether treatments work or not, because doctors and other health professionals are trained to try and select the right treatment for the right person. And that means that if you then just simply compare people who were prescribed a given treatment with people who were not, it's not a fair comparison, because the people who got the treatment have been selected to have it, and the people who weren't given the treatment were selected not to have it. And those reasons why the treatment was selected or not are likely to be very important. And therefore, they will introduce bias into the comparison. We often describe this as comparing apples with oranges. So if there's this particular treatment that you know is particularly good for apples, then you'd expect that you'd end up with more apples in the people given treatment, and you'd end up with fewer apples and more oranges in the people not given treatment. So you're not comparing like with like. If all you do is compare people given a treatment versus people not. If instead we randomly allocate people to whether they receive treatments or not, in other words, you decide whether that or not they get treatment on the toss of a coin, something which is completely unpredictable to the medical team looking after them, then you end up with two groups of people who are exactly the same except for whether they receive the treatment or not. So you can see in the picture below, and we now have the same proportion of apples and oranges in both groups. So those two groups are exactly the same, apart from whether one group was given treatment and one group was not given treatment. And then when we follow up those people to see how they do, and if we see differences in what happens to them, we can infer that those differences are due to the treatment, because in all other respects, the groups should be the same. So that's one of the mistakes that we can make, which is systematic error. And that's why randomization is so essential, because it is the only way to reliably remove systematic error. The other sort of error we can make is random error, or the play of chance. So I just want you to imagine an experiment in which you're asked to toss a coin to see whether a patient gets better. If the coin lands up heads, the patient gets better. If the coin lands tails up, the patient doesn't get better. You are assigned to be the treatment arm. So you're asked to toss the coin 10 times for the 10 patients who were given treatment in your trial. You have a colleague sitting next to you who's given an exactly identical coin and asked to do the same thing, except when they're tossing the coin 10 times, they're tossing the coin for the 10 patients who were not given treatment, who are randomly allocated not to be given treatment in your trial. So you've got two coins, each tossed 10 times. Now, if you got 7 heads and your colleague got 3 heads, would you be very surprised? And I think we know that no, we wouldn't be that surprised. When we toss a coin, we expect to get heads about half the time, but we know that we won't get it half the time precisely. And especially if we only toss it a relatively small number of times, getting an imbalance like 7, you getting it 7 heads and your colleague getting 3 heads isn't very surprising. But if you then put that into a trial result where you said, well, 70% of people got better on treatment and only 30% of people who didn't get treatment got better, then you could misunderstand that and think that actually the treatment is really, really good. Whereas in fact, all it is the play of chance. Now, we can avoid random error by randomizing very large numbers of people. Because if you were asked to toss your coin 10,000 times, and so was your colleague, then it would be incredibly unlikely that you would get 7,000 heads and your colleague only get 3,000 heads. You would know that there was something going on there. So that's why you need really large numbers of people in trials. And the reason that it's so important that we minimize these errors, both systematic error and random error, is because most treatments that we have in medicine nowadays, this might sound slightly pessimistic, but unfortunately it is true, but actually not a reason for pessimism, is that most treatments we have only have moderate treatment effects. So they only reduce the risk of a bad outcome by a modest amount, say by about 20%. So rather than 10 out of 100 people dying, for example, a good treatment might reduce that to 8 out of 100 people dying. So that's a 20% reduction from 10 to 8. And because those treatment effects are only moderate, we need to make sure that any errors in our experiment are really negligible in comparison to the size of that effective treatment. So any systematic error that we have could easily obscure that real effect. Any random error that we have might obscure that effect or might make an effect appear when it's actually not real. So it's really key that we avoid those errors as far as possible by randomizing people and by randomizing suitably large numbers of people. And this is something in which the Population Health Research Unit that I work in has really pioneered over the last 40 years the methods of making trials like this possible. So a new disease that we're all over familiar with now emerged really came to public attention in 2020, caused by a coronavirus called SARS-CoV-2, and the disease is called COVID-19 because the virus was actually identified right at the end of 2019. And it's a disease which has multiple phases. The majority of people who get infected get a viral infection, a bit like a bad cold or flu, and that resolves, and most people get better of their own accord. But in some people, the disease progresses and becomes more than just an upper respiratory tract, so sort of head and throat infection, becomes a pulmonary or pneumonia infection. And those people tend to get more ill, and a larger proportion of those need to go to hospital for help. Some of those people, the disease then triggers an exaggerated host inflammatory response. So this is the response of the body's own immune system to try and deal with this viral infection, then becomes exaggerated, if you like, and then can actually become a bad thing in its own right, that there's overwhelming inflammation going on. And many of these people get very sick in hospital. And unfortunately, a sizable proportion of these people, especially early on in the pandemic, died from the condition. But this means that we have a disease which has multiple different potential treatments. We could try giving people treatments to kill the virus early on, and maybe they would still work later on in infection. Or we could give people treatments that dampen down their own immune response to the virus to try and make sure that it isn't an exaggerated response, but it doesn't do them harm. So these are the different treatments potential treatment options that we had. But back at the beginning of 2020, nobody knew whether any treatments would work. And there was huge amount of interest and a huge public health need, because if you ended up in hospital, then about 1 in 4 or 1 in 5 people sadly died of COVID-19. So it was, as we all know, causing a very large number of deaths in the UK and around the world. So recovery was set up early in March 2020 as the pandemic arrived in the UK by Peter Horby and Martin Landray, who were the chief investigators. And there's a picture of a London bus on the slide because their meeting was actually largely due to almost coincidence when Martin Landray met a colleague of Peter Horby's on that bus as they were both travelling to the Wellcome Institute in London. They drew together a team of experienced trialists within the MRC Population Health Research Unit and associated groups that managed to take the trial from protocol to the first patient into the trial within 9 days. If any of you are familiar with running trials, you'll know that usually going from a finished protocol to first patient would typically take 9 months to a year. But clearly we didn't have that long. Now, recovery, like all trials, is really team science. So although the chief investigators in the middle of the sort of familiar public faces of the trial, they drew together a team within the Population Health Research Unit and the university department in which we sit, of trial managers, IT experts, data management, statisticians, clinical support to work with them to help deliver the trial. And in turn, we were then supported with a lot of help from the NIHR's Clinical Research Network to recruit sites, so hospitals where patients were being admitted, which each hospital needed its own team, headed up by a principal investigator, but an own team of staff to recruit and look after the patients going into that hospital and going into the trial. So it's a huge effort. We were helped by as I say, the Clinical Research Network to identify sites. But to get started, each site needed a number of approvals. I'm just going to start this video running, which shows you the sites as they opened up. You can see the number of sites opening on this panel on the right-hand side. And then as the green dots emerge, it shows you the numbers of patients that they are recruiting. But before any of those sites could start recruiting, they needed to have all sorts of approvals. They needed study drug to be supplied, and they needed trained staff. But in the first 100 days, as you can see here, we recruited over 10,000 patients. In fact, we recruited 10,000 patients in about 8 weeks. So recruitment was astonishingly fast due to an incredible effort of the team, both in Oxford, but in particular at the sites, who were overwhelmingly enthusiastic to help because they could understand the public health emergency that was going on at the time. Trials also need participants. Now, in the interest of time, I won't play you this video, but you can view a video which, for about 2 minutes, gives the information that participants were shown when they were asked to consider the trial. This can be viewed on our trial website, recoverytrial.net. But this shows you the sorts of people that the sites were recruiting into the trial, and so they actually match very, very nicely the sorts of patients being admitted in the hospitals of the UK. And overall, we have recruited about 10% of all patients admitted to hospital in the UK with COVID-19 were recruited into recovery. And they look to be a very representative sample. So the average age was about 60, nearly 2/3 were men. The ethnic breakdown matches that of the population being admitted to hospital. They'd been unwell for about 9 days before they ended up in hospital, and some of them were in hospital but weren't poorly enough to require oxygen, but the majority of these patients did need oxygen in some form, and about 10% actually needed a ventilator, so were on a breathing machine at the point at which they were randomized. And they were a fairly poorly group of people, so over half of them had some form of major comorbidity listed at the bottom of the slide, but they had some other major problem like diabetes or heart disease. And again, we knew that this was matched what was going on in the NHS at the time. So the trial was moving very quickly, as I've said, recruiting 10,000 people in 8 weeks and continuing to recruit. And we had a data monitoring committee. So that was an independent group of experts who reviewed the data as they emerged during the trial. So people running the trial here in Oxford, we didn't know what the results were as things went along. But that was the job of the independent data monitoring committee, whose primary responsibility was to maintain the safety of participants in the trial. And that if the trial results were going to be made public, that they should be informative and useful for doctors looking after similar patients. And so the first comparison that we actually ended up stopping was testing a drug called hydroxychloroquine, which was a drug popularized by Donald Trump, who thought that it was the bee's knees and a very good treatment to have. But we randomized about 4,500 people to either receive hydroxychloroquine or not. And we were told by our data monitoring committee to stop the recruitment because they saw no realistic prospect that hydroxychloroquine was going to show benefit. This graph is what's called a Kaplan-Meier survival curve. It shows time along the bottom, and it shows the proportion of people dying on the y-axis up here on the left. And as you can see, the proportion of people who were dying who were in the hydroxychloroquine group was slightly higher than the proportion of people dying in the usual care alone, the control group. And although we didn't prove beyond doubt that hydroxychloroquine was bad, there was very little prospect, or virtually no prospect, of this hydroxychloroquine ever being proved to have a meaningful benefit. So the Data Monitoring Committee suggested that we stop recruiting to this comparison. And that led to a rapid change in clinical practice. So hydroxychloroquine was a very popular drug around the world, being used widely, particularly in the US, where their drug regulator, the FDA, had given an emergency license to allow doctors to use hydroxychloroquine for patients. But they revoked that license on the 15th of June. So we started our trial in the middle of March. We announced these results on the 5th of June, so less than 2 months later. And on the 15th of June, the FDA revoked their authorization and the use of this drug around the world plummeted. So this was the first practice-changing result from recovery. Not necessarily the result anybody hoped for. I think we'd all liked our first result to be a benefit, to show a drug that worked. But actually, showing drugs that don't work can be equally important. Sometimes drugs might have side effects, and it's important not to expose people to side effects if they have no benefit. But it's also important just to not distract doctors and nurses and other healthcare staff from trying to deliver a treatment which isn't worthwhile. It'd be better investing their efforts elsewhere. But not long after that result came our second result, which was dexamethasone, which is the result that lots of people have heard about with recovery. On the left-hand side here, you can actually see 4 Kaplan-Meier plots. For the one in the top left shows what happens with all participants. And I'm sorry the numbers are a bit small. There was a lot of information to try and fit on here. But it showed that overall dexamethasone reduced the risk of death by about 1/6. But what was actually the hidden message behind that was that it really mattered how ill the patient was as to what benefit they got. So for people who were very ill, for people on ventilators, so these typically people on intensive care units, on breathing machines, dexamethasone reduced the risk of death by about 1/3. So it reduced the risk of dying from about 40% down to about 25%, which is actually a very big effect, meaning for every 8 patients that were treated with dexamethasone, 1 life was saved, which was quite astonishing. Patients who were needed oxygen but didn't need a ventilator also benefited substantially. Their risk of death was reduced by about 1/5. For patients who were not ill enough to need any oxygen, actually dexamethasone wasn't beneficial. You can see here that, like with the hydroxychloroquine comparison, the red line, the dexamethasone line, is above the usual care line, showing no real benefit for dexamethasone. So dexamethasone is not recommended for people who don't need oxygen, but that is a relatively small number of people admitted to hospital nowadays with COVID. But on the same day that we made these results public, the chief medical officers in England released this alert to all doctors in the UK that dexamethasone has been demonstrated to have a clear place in the management of hospitalised patients with COVID-19. And guidance was given immediately for patients who needed oxygen, in particular those on ventilators, should be treated with dexamethasone. So this happened 87 days after the trial started, and overnight there was a change of practice in the UK, with dexamethasone becoming widely used. And there have been estimates made by others that the use of dexamethasone has saved about 1 million lives since these results have come out. Recovery's tested a lot of other treatments and got a lot of other, what some people call negative results, but these are all important negative results, a bit like the hydroxychloroquine results, because drugs like lopinavir and azithromycin were being widely recommended by guidelines, either national or international guidelines, as potential treatments for COVID-19, but we showed that they did not work. Convalescent plasma is an interesting example, and I show you the Kaplan-Meier plot here, where you can see that there is absolutely no effect for convalescent plasma here, with the treatments and the control groups looking almost identical. Because in the UK, when convalescent plasma was considered, this is plasma that's been collected from people who've recovered from COVID, which therefore contains antibodies against COVID. And you can then give it back to other patients who have just got fresh COVID, if you like, to see whether giving them those antibodies helps them. So that's what convalescent plasma means. In the UK, when this treatment became available and the NHS Blood and Transfusion Service started to collect this plasma, it was decided the best thing to do is to put it into a trial like Recovery and another trial called REMAP-CAP. In the US, they took a different approach. They decided that randomization wasn't feasible, and they spent hundreds of millions of dollars giving convalescent plasma to people, unaware of whether it was doing patients any good or not. So they spent hundreds of millions of dollars doing that. The UK spent a few £1 million doing a trial which randomized over 11,000 people and showed that, unfortunately, convalescent plasma was of no benefit and therefore should not become part of standard care. And I think that's a really important difference in approach. And the UK has informed the world about the utility of convalescent plasma. And other treatments which we've tested, which have also been shown not to work, are colchicine and aspirin. And they were important because they're very, very cheap. They're widely available throughout the world and very, very cheap. There were good reasons to think they might work. Unfortunately, they didn't. If they had worked, then they could have transformed the treatment of COVID, not only in the UK, but around the world. And so this slide sort of shows the progress of recovery. We're over 2 years now since the trial started, and it shows the various landmarks that we've had, including another treatment called tocilizumab, which we found, which is a powerful antibody-based treatment to reduce inflammation, which we showed could reduce death. And then most recently, earlier this year, we've shown a different form of antibody treatment. So not convalescent plasma, but purified antibodies made in a laboratory could benefit certain types of patients. And then most recently, just last month, we showed that a drug called baricitinib, another anti-inflammatory drug, could also reduce death. So overall, Recovery's found 4 treatments now which can reduce the risk of death in patients admitted to hospital. So what's going on with the trial now, just as I close? Well, COVID-19 is a pandemic, which means it's a worldwide problem and so it needs worldwide solutions. And so the trial is now running in different countries, shown in various colours on this slide. And the advantage with that is that we know that the pandemic goes through different stages at different times in different countries. So there are waves of infection. The UK had a wave before the US. For example, Italy had it before the UK and so forth. And we know that, for example, China is in the middle of another wave now. So if you have an international trial, then it means that the trial can continue to recruit, even if things go relatively quiet in one country. Hopefully you can continue to recruit in another. And it also allows us to transfer the knowledge that we've generated from running the recovery trial to other trials groups around the world. So we've got 5 treatments in the trial at the moment. We're continuing to test therapies, as I say, directed against the host, so directed against that exaggerated inflammatory response. So we've shown that dexamethasone works, and that was a low dose of dexamethasone. And so we're now asking the question, well, is more steroids better? A very reasonable question. And so we're comparing high dose dexamethasone with the standard dose, and we're testing a drug called empagliflozin, which is actually a treatment for diabetes. But there is some evidence to suggest that drugs like this might be beneficial. And then now that we're 2 years on after the virus is discovered, there are more novel antiviral therapies available. I mentioned one very briefly, these monoclonal antibodies that we've already shown work in some patients, but unfortunately the virus can evolve and does and can become resistant to these monoclonal antibodies. So the Omicron variant in circulation at the moment is resistant to the monoclonal antibody therapy that we showed worked predominantly against alpha and a bit against delta. And so we're now testing a new one called sotrovimab. And there are other antiviral treatments, tablet treatments, which interfere with the virus in different ways, called molnupiravir and nirmatrelvir, which are also now in recovery. These latter two, in fact, all of these antiviral treatments, we know they work in people early in infection who aren't in hospital, but there is substantial uncertainty about whether they work for the patients who end up being admitted to hospital, which in one way are particularly important because that is the group of people who unfortunately die, people who end up in hospital. So we need to know whether these treatments work in that situation as well. And so we've learned a lot from recovery. we've learned that you can embed high-quality trials into routine clinical care and get benefits for both. Not only do you get trial results out of it, but you also help routine clinical care help it to focus its efforts so that it doesn't have to waste time delivering treatments which may or may not be effective, but can focus its efforts on those which are or maybe, in other words, are in a trial. We've made the trial as transparent as we can, so we've put all of our trial documents on our website, so you can go there and you can see every version of the protocol that's ever been used, all of the documents submitted to the ethics committee and their response. They're all there on the website, so everybody knows what's going on. And one of the best bits about recovery has been the collaboration. So we have a team in Oxford, but they're a fantastic group of local investigators at hospitals around the NHS in the UK and now internationally. And it's been great that we've been able to acknowledge all of those investigators, over 7,000 of them, including all of their staff at their hospitals who've contributed to the study. And we acknowledge them in all of the publications that we make. And recovery has worked very well in a pandemic situation, but actually this type of trial that it is and the methods that we've used are widely applicable, and they go beyond COVID-19 or infectious diseases. And hopefully many of the lessons that we've learned from recovery can be applied to diseases of other common conditions, which even in quotes normal times without a pandemic cause a lot of morbidity and mortality in the UK and internationally. We need more trials in all of these conditions, and we hope that some of the lessons we've learned from recovery can be applied there. So I'll finish now, and just here are some of the people that the trial we need to say thank you to, including our funders, many of the UK organisations that supported us. As I say to all the doctors and research staff in the NHS hospitals and internationally, but most importantly, we must thank the patients who at a particularly terrifying time in their lives were brave enough to volunteer to take part into a trial like this, without whom we wouldn't have any of the information that we do have. So we are very, very grateful to them as well. But thank you very much for your attention, and I'm happy to take any questions.